Greig is a transplanted East Coaster. He did an ecology bachelor at Guelph and continued to do some training in GIS, and that led to a master's in spatial optimization about river restoration at Dalhousie. Actually, it was a master's with a scope and content of a PhD. I had the pleasure of looking through it when he joined, before he joined here at UBC. He went on to work for 5 years for DFO here in Vancouver as a spatial planner, working notably on the spatial planning process for Central Coast, heavily involved in that. And he joined then UBC 2 years ago to do a PhD in cooperation with the Pacific Salmon Foundation. It's about decline of salmon in the state of Georgia, and he'll be talking much more about some interesting aspects of that work. It actually took quite a while to get him on board here at PhD. He was interested, but also had some good work at DFO, but he managed to get study leave from DFO, so thinking is that he will be returning to DFO after this. And what else can I say about Greig? Some keywords would be he likes data science, how to deal with data, geospatial analysis, data visualizations, programming, database design, statistics design of various kinds. So it fits pretty much into this overall quality topic for our seminar series here. And today he'll be talking about modeling long-term ecosystem and productivity changes of the Georgia Strait, as you can see from the this first slide here. And please join me in welcome Greig to give this talk. And let me just mention that this talk is being recorded. So if you don't want to be seen, keep your cameras off when you get to the questioning part. But welcome, Greig.
Thanks very much, Philly. Thank you, everybody, for joining today. I'm really looking forward to talking about the research I've been involved with in Vili's lab. And I've been lucky enough to be part of the Salish Sea Marine Survival Project now for a bit over 2 years. And it's a great collaboration. And so I'm looking forward to sharing a bit of background today on that project. I'm going to talk about the modeling work that we're doing in support of the Salish Sea Marine Survival Project. And I've been involved in that with numerous collaborators. So in addition to Vili, my committee includes Brian Hunt, Carl Walters, and Isabel Pearsall, all of whom I'm indebted to, so thank you. And there have been numerous other collaborators along the journey. So Vijay Kumar, Joe Buskowski, Jeroen Steenbeek, Fanny Couture, many others that have provided support. And then I'd be remiss not to mention the support of DFO, the Pacific Salmon Foundation, MyTex, NSERC, and Ecopath, the Ecopath Consortium. So let's see, I'll start out by giving you some general context for this work. And then I'll get into specific research questions we're focusing on related to productivity of the pelagic zone in the Strait. I'm going to proceed assuming that all of you are quantitative modelers. Then I'll get into, but I guess I won't assume that you know more than I did a couple of years ago about details of the local ecology of the Strait or the biogeochemistry and hydrodynamic characteristics and the changes that have occurred. So I'll take some time to review that. And I hope by the end of the talk today, you'll know a bit more about that than you did about the SSMSP and the Strait of Georgia, and also the specific modeling approaches we're using. And if there's time, I hope I can get a chance to talk about some of the emerging challenges I feel are probably common for a lot of ecological modelers, like challenges with data access and integration. And yeah, if I can, I'll try to share I'll talk about some tools that I've found to be quite helpful so far. So Chinook, coho, and steelhead smolt to adult survival in the Salish Sea region has declined substantially over the past 30 years. When compared to survival and abundance trends, when survival and abundance trends are compared to those outside the strait, there's evidence that an effect on survival is occurring in the Salish Sea. So this smolt adult survival, or SAR, or marine survival, for many stocks seems to have declined an order of magnitude compared to what they experienced in the 1970s and the early 1980s. The following trend shown here, the following trend is from analysis of coded wire tag data of wild and hatchery fish, but mostly hatchery fish. The coho exhibits a clear declining trend beginning in the early 1980s. And but prior to that, the survival data are sparse because the coded wire tag program really didn't ramp up until the late 1970s, early 1980s. The results of the Ruff and et al. Analysis of the Chinook survival indicates a finer scale variation at the individual. I see there's somebody in the waiting room, but I guess Catherine, you'll take care of that. Okay. Yeah, so that the finer scale variation of the individual population of Chinook compared with coho is something that researchers have found. And so there's a weaker coherence in the survival trends among populations within the Salish Sea versus outside the Salish Sea. Sorry, I got that reversed. There's more coherence within the Salish Sea. And Kilduff and others in 2014 found what appeared to be a common pattern among populations coastwide. So they found declining survival in Chinook between 1985 and 1995. So this has had some very serious impacts on everything from the ecosystem to fisheries to the economy to treaty rights and to the way of life of many of the communities locally. For example, the Strait of Georgia Chinook and coho fisheries were once among the most valuable in Canada. So the steep drop in both catch and effort shown here in the BC Southern Coast recreational fisheries illustrates this major impact. Note how the falling effort, the effort fell along with catch. And this wasn't due to fisheries closures or regulation, but those did come subsequently. Beginning in 1997, DFO implemented a number of fishery management measures to reduce harvest impacts on coho, and more severe measures were implemented subsequently. And eventually the recreational fisheries were mostly closed in the strait. So I'm going to talk a little bit now about the Salish Sea Marine Survival Program. I'll just call that the SSMSP. Salmon recovery efforts up till the late 1990s, they were mostly focused on harvest, on freshwater, on estuary impacts and hatchery impacts. But between 2000 and 2010, it became apparent we needed more research devoted to changes in the marine environment as new evidence came forward. So the forerunner of the SSMSP was the Strait of Georgia Ecosystem Research Initiative, which documented major changes in the Strait of Georgia since the early 1980s. However, through to 2010, there were no comprehensive studies of mechanisms that were regulating the production of Chinook and coho and steelhead salmon in the Salish Sea. So that led the Pacific Salmon Foundation and the US-based Long Live the Kings to partner under the banner of the SSMSP. In 2012, they co-launched the project, which involved extensive partnerships with something like 60 institutional partners. And the main goal was to try to separate the effects of multiple factors and to try to identify the primary determinants of salmon survival and then what mitigation measures might be possible. The research I'm talking about today falls under the trends and modeling category, and that's kind of broadly where Vili's group fits in. In this category, we look at existing and new data that are brought together to analyze and to model relationships between salmon and their ecosystem. And we particularly are interested in the cumulative effects of multiple factors and to build back to factors ultimately driving survival down over time and holding survival down. So where other studies looked at short-term productivity patterns, we're looking at decadal scale patterns going back 40 years. We are trying to evaluate the relationship between long-term changes in environmental productivity and the productivity of higher trophic levels. And so we've set out to develop a coupled hydrographic and biogeochemical model of the Salish Sea and link this to a spatial food web model in order to evaluate how the combination of changes has affected productivity, food web structure, human, and how human impacts have affected marine survival. So this involves a large amount of data assimilation, which I'll talk about in a bit, and large quantities of, for example, large quantities of environmental, biological, ecological fisheries data. So broadly speaking, the SSMSP created hypothesis categories to try to organize their research efforts a bit. And the primary hypotheses were lumped into either bottom-up processes, top-down processes, and additional sort of additional factors processes. Now, the bottom-up processes include things like weather, water conditions, productivity, whatever bottom-up factors determine prey to available juvenile salmon, and it can include consideration of competition-related hypotheses as well. So that's the bottom-up processes are the main ones I'll focus on today for this talk, although we are exploring and interested in certainly in the top-down processes like predation or vulnerability to predation and how that affects survival. For those of you who aren't familiar, I'll just take a second here to review, the precise area we're looking at or the rough area we're looking at. The Salish Sea is approximately 18,000 square kilometers. It's nested between Washington State and British Columbia. There are three major water bodies: the Ecospace Strait, Puget Sound, and the Strait of Georgia. And there's also actually the Harrow Strait and San Juan Islands area sort of in between. There's about 8 million people right now that live in the surrounding coastal area, and that number has been growing very quickly. And when we talk about the Strait of Georgia here on the right, we shouldn't forget to talk about the Fraser River, because the Fraser River is very huge compared to other rivers that drain into the Salish Sea. The Fraser is responsible largely for the estuarine conditions of the Strait of Georgia and the whole sea. It contributes probably 50% roughly of the freshwater entering the sea. And the residence time, it causes the residence time of the total volume of the sea to be surprisingly low. It's ballpark 1 year or less, according to one source that I found. And here, Rich Palowich has great diagram that summarizes the estuarine circulation that occurs. So that helps keep the waters nutrient rich and is responsible for some of the high biodiversity that we see here. So I'm going to talk a little bit about the two species that we are focused on, coho and chinook. All of the salmon life cycles here take them from rivers to estuaries and then through thousands of kilometers of ocean habitat before they return to their natal rivers to spawn and start all over again. So here's a quick primer on the two species that we're interested in. There's a lot we don't know about the distribution and the migration of coho and chinook, especially as juveniles, though recent tagging and tracking research is helping. So one thing that we know differentiates coho and chinook from these other species is residence time in the estuary and immediately upon ocean entry. Most, but not all, the sub-yearling chinook juveniles remain in proximity to their natal rivers and during the first summer and fall, so 100 to a few hundred kilometers from where they entered the ocean. Then they move north during the winter and the following spring. So most, but not all, juvenile chinook in the Strait of Georgia remain close, regardless of their freshwater history, whether they're sub-yearling or yearling or ocean or stream type. In contrast, the sockeye, sockeye, chum, and pink are not usually found in the coastal waters, such as Puget Sound and Strait of Georgia, although chum do use the estuary along with beside coho and chinook in the early days of ocean entry. The migration is generally characterized by directed movement northward for the pink, chum, and sockeye species. And then they distribute into the offshore waters eventually until they mature. In terms of diet, coho and chinook have a tendency to be piscivorous more than the other species. Rapid growth upon ocean entry to get big enough to eat other fish may be an important strategy for chinook especially. Several studies have found that chinook had the greatest tendency to eat fish. Here we see some of the trawl survey results from TFO trawl surveys that Beamish and Neville and others have been involved in. And we see relatively high proportion of teleosts and fish in the diets of coho and chinook. And that varies year to year. There's a typo, sorry, chinook. But we can go back a little further. So the biologists argue in 1986 found chinook and coho from Cowichan Bay fed heavily on herring between July and September, with like 90% of the stomach content by weight. And that's a diet study, a series of diet studies from the 1970s. And then based on the index of relative importance, decapod larvae were the second most important item in the diet of these species in Cowichan Bay. I have a bit of a work in progress. There'll be a couple of these I'd like to just briefly show you, just to break things up a bit. So here's a little wonky visual of some of the diet data that was rescued from in an effort by Osgood and others and McKinnell and Perry in 2016. So this is data from the 1960s, and it's historical diets of forage fish and juvenile Pacific salmon. So the results show the trophic linkages and between the juvenile fish on the right and their prey on the left. As I said, this is a bit of a work in progress and it's somewhat wonky because you probably will notice there's no coho showing up here, which is too bad. But So there's pretty apparent differences that you can see just by exploring the data like this. We see that, interestingly, insects made up a larger portion in terms of frequency of occurrence in the diet of Chinook back then, and that's compared to today. It doesn't seem to be the same. It may be some bias in sampling, but it may be a true trend. We did. I made a bit of effort to try to estimate the biomass, the proportion of diet in terms of biomass that is represented by these different prey items. And that reveals that fish catching a fish can mean a lot. So that does seem to be reflected in the diet data from the 1960s, that fish were important in the diet. And that includes juvenile fish and fish like oolakan. So, oh, right. So this is an interesting study as well. I forgot I was going to mention. The Chittenden study looked at really small Chinook, those that are sampled from the estuary and the bay. And they found that larval fish did play a bit of a role in the diet, as well as bigger fish, and then the others were not really much of a surprise. So getting into some of these changes that we're going to explore with our model, the changes that have happened particularly to influence productivity, we believe, have to do with ecology and with climate and physical chemical changes. So as I was saying, the forage fish play an important role in diets. We know that. Daly and others have found, several studies have found that during years of lower survival, much of the variance in marine survival of Columbia River Chinook and coho was related to the biomass of ichthyoplankton off the coast. We know that herring play an important role in diets, but timeshares of the young of year herring don't really go back far enough to look at the time horizon that we're looking at. The Strait of Georgia stock assessments of spawning stock biomass don't really indicate an overall biomass trend that jumps out for the time period of interest. Although there's some trend maybe that we can extrapolate or get at if we squint. But there is a strong relationship observed between juvenile Chinook salmon size and consumption of herring that may have important consequences or implications. We also do know that herring could play a buffering role on predation on salmon, on juvenile salmon by their predators that would otherwise eat salmon. So if there's a lot of herring, we see in Surma's diagram here how important herring is in the Pacific ecosystem. Although, that said, some juvenile salmon end up incidentally eaten when they're in the wrong place at the wrong time around schools of herring. So that one's a tough one. Something that is important is that spawning dates have become earlier and spatial temporal changes have been observed in the Strait of Georgia. Chinook salmon could be dependent on a later spawning of herring as prey due to their presumed smaller size. Oolakan, I'll just pause here for a second. So this figure on the right is the difference in spawning date from Bolt and others 2018. So it shows a gradually earlier date of spawning in the Strait. And then the work of Theriault here in 2009 is showing spatial changes that appear to have occurred in the spawning stock biomass. So a concentration of the spawning stock biomass in the northwest and a decline of spawning stocks on the mainland shore. Although in recent years that may have changed. Oolakan is another forage fish that's really interesting to look at because they did appear in the diets back in the 60s and 70s of Chinook especially. There's not a lot known about the Fraser River Oolakan, although Murdoch recently did work on some stock assessment, on a stock assessment. I didn't have that handy, so I haven't put the figures in there, Murdoch. But historically, it seems to have been a much higher stock size, and we see that Oolakan in the 60s and 70s was quite prevalent. The COC Marine Survival Project researchers are also studying spatial and temporal changes in zooplankton assemblages, biomass, and abundance. And they've assessed the relationship between those changes and marine survival of coho and Chinook. The plankton food web is very complex. So here's a food web model of the coastal areas in the Wanafuka and northern Puget Sound. This model was a result of several years of work, ecological synthesis effort by a team at the University of Washington. But since then, much more detail has been uncovered. Plankton communities in the Strait have changed during the past 50 years in terms of species compositions, number, size, and timing and duration of blooms. Cold water copepod species have been declining in the southern part of their range. Jellyfish and crab larvae are increasing. In the Strait of Georgia, abundance of the largest copepod, Neocalanus plumcus, has occurred over the past 15 years, and the timing of peak production has shifted 50 days earlier. SSMSP researchers have also found that the Pacific Decadal Oscillation, sea surface salinity, and the modeled start date of the spring phytoplankton bloom in the Strait also correlated with zooplankton trends. They identified two time periods, 1996 to 2006 and 2007 to 2018. These were found to represent a period of declining and a period of increasing zooplankton biomass anomalies, respectively. But again, the time series don't really go back to the early 1980s. So it's helpful, it's interesting, but it really doesn't give us as much information as we'd like about the time period we're really interested in. The past spring, I did a large lit review to try to understand what the important pathways are, and I was concerned with trying to figure out how the linkage could be reasonably made between the biogeochemical, so the plankton, the lowest trophic levels in the chemistry of the Strait, and the upper trophic levels. And there's not time today to go into further detail on that, but it's very complicated and there's a lot that's unknown. This is the seasonal life cycle and development of copepods. Generally, the life cycle illustrated here by Makis and others in 1998. And but not a lot is known about what triggers the emergence of the copepods in the spring. So I'm going to move on to climate, hydrodynamic, and chemical changes in the Strait. This is a helpful diagram, I think, and it shows the major processes that collectively control bloom timing. And many of them have been changing due to climate change and other anthropogenic pressures. I'm just going to talk briefly about a couple of important ones, the ones that we feel important today or important for our modeling work. The winds in the estuarine circulation replenish nutrients in the surface layer. The freshwater discharge brings suspended particulates and promotes stratification, and sunlight provides energy for photosynthesis. So looking at some changes observed or predicted in the long term, we're expecting changes in the flow pattern, discharge volume pattern of the Fraser River. Researchers think that over time, it'll become more like a river with flows that are dominated by precipitation patterns similar to other rivers that drain into the Strait of Georgia. But at the moment, it's dominated by snowmelt, with a very steep spike in peak summer freshet. So when I look into the data, the discharge data from Hope, don't see a major shift in the time period of interest for us. Although what is happening is consistent. What's shown in this figure on the right is consistent with what others researchers have found. So a slightly steeper spike associated with sort of heavier snowmelt a little later in the year. This higher peak discharge may be associated with increased harmful algae blooms, although how those would be affecting survival is postulated, but not really clear. Winds, though, are where things get really interesting. So when I think about winds and their influence on the bloom timing of the strait, I think of it as kind of like a Goldilocks productivity situation. So if you have not enough wind, you don't get mixing that brings nutrients to the surface for primary productivity. If you have too much wind, if it's especially early in the spring, they can disrupt the spring bloom or delay it. So there's really an optimal environmental window there. So as the winds increase, the reason the winds affect things is that they increase the mixing layer depth, they bring nutrients to the surface, and almost every decrease in the biophysical model, of the Collins, Allen, and Palowich 2009 model, the bloom timing model, was associated with strong wind. They are generally agreed to, strong winds are generally agreed now to delay the spring bloom if but only if they occur early in the year. If they happen after the first spring bloom or throughout the summer, and they're sort of episodic, they can sort of reignite the bloom in the strait. So that's some important dynamics. So there have been changes in wind, and we downloaded wind from the European Commission's retrospective model, which extends back to 1980, the ERFA5 Copernicus model. And Carl Walters calculated mean monthly wind velocity along the Georgia Strait main axis. So positive for southeast winds and negative for northwesterlies, winds from the northwest. When you looked at just the springtime average of this index that he created, he found a negative trend with lots of year-to-year variation, but it kind of correlates with coho marine survival rate declines and the changes in the proportion of coho rearing in the strait. So this is a spring northern transport index, pictured on the left, and the moving average highlighted in the bright red. So if the southeasterlies cause converged plankton concentrations in the northern strait, then there could be less such food concentrated by wind in more recent years in the northern strait. But the relationship is not, clearly convincing, but it seems to explain some of the things that have been seeing, like we've been seeing, like a big drop in the early 1990s in coho in the strait. But you can note also that the plot of the cumulative transport, so that's velocities summed over all months, shows upward trend. So for all years except for a brief period in the late 80s when the northwesterlies were strong and persistent enough to generate net southward transport. And Carl remembers when this, when windsurfing took off at Jericho Beach because of this nice western wind that came along the beach for much of the summer. So again here, I'm going to just jump out. Please let me know if you have trouble seeing anything here that I'm going to try to share. I'm going to Pan through here. I'm going to just visit a demo here that I am trying to put together. It's 1998 there. Let's just find 1980. Nope. Wait a sec. So organized. Okay, so this is a tool I adapted to try to help, because if you're like me, then it can be helpful to have more than the visuals I just showed you to really kind of get a feel for what's going on. This is a JavaScript-based simulation of wind from the ERA5 Copernicus data. And the data are interpolated onto an image, and then tens of thousands or a varying number of particles that you can kind of play with here are dropped onto the image, so that basically simulating through this great this particle simulation of the direction of wind and the magnitude. So 1980 is kind of an archetype of the prevailing wind patterns of the Pacific Northeast. We're looking at 1980 January here. We see prevailingly southeasterly winds. This is this is a wind that promotes downwelling, the wind direction. So the wind is headed north, kind of towards the Aleutian low. If we progress into February, we see the winds strengthening in this year, but generally the same pattern. And then in March, there's this dramatic shift. So this is a spring transition, and this transition you see here can promote bloom of phytoplankton in the Strait of Georgia, which is pictured in the center here. The shift can cause conditions with respect to wind to become a bit calmer in the strait. And so this is happening in March in this year. But if we compare it to sort of a less archetypical year, say 1998, I'm going to just put 1998 on the right screen, and we're looking at February, and we see February, okay, seems fairly similar. I'll move it to March. Let's just figure this out. The winds are not quite as strong. I'm going to move into April here. And now it's pretty clear I'm overwhelming my GPU. Anyway, so you can see the wind shifting. I'm just going to turn off this one on the right. And let's take a look at what the summer conditions are like in the Strait. So the summer, August of 1998 seems a bit calmer in the Strait, but we see how the wind in the summer comes from the northwest, and it promotes upwelling. It comes into the Strait through the Juan de Fuca, it kind of twists around and then can turn northward. It can start to turn totally north, in fact, and transport surface waters to the north of the Strait, and sometimes the opposite pattern can occur too. And what I was finding was that 1998 was kind of a variable year. It didn't seem to conform. So let's just jump back to the main presentation here. So our research approach, I'll just talk about that very briefly. Why do we model anything? Why are we doing this? I think that models are really helpful to understand which processes are important, which are not, but they're also important from a lot of other reasons too. A modeler like myself learns a lot about the system, for example. And the huge spreadsheet on the right is an idea of, or it gives you an idea of what we go through when we're not familiar with the system, we don't really know what's important and what's not. So this is a huge spreadsheet I put together in the first 8 months of being at UBC. It's just a portion of the spreadsheet, and the long list on the left is things like parameters, like diets and other sources for different species and functional groups. And, just to keep track of things, I've got the figures from these sources on the right. But a model is really helpful at helping us figure out what is important and what's not. I think that's a really big reason why we're doing this modeling work in this context. It's also a helpful way to synthesize and collate data and to explicitly evaluate alternative hypotheses. So in this scientific context, that's really what we're aiming to do, and that's what I'm going to talk about today. We're talking about using mechanical models, not statistical models, not correlative models, but mechanical models to help complement those types of models. We're trying to keep the models simple, but not simplistic. And we're talking about using multi-species models, ecosystem models, and we're using software, the Ecopath with Ecosim and Ecospace software that Vili and Carl and others have been developing at UBC for a long time. The task I have and collaborators have is to introduce hydrodynamics and biogeochemical processes and modeling to the EWE or Ecopath, the Ecosim and Ecospace framework. I'm going to just pause here to give you a good example. I think what is a good example of a simple model, the Collins, Allen, and Palowich predictive model of bloom timing from 2009 that they've subsequently continued to work on. But I thought this was really interesting. It's a one-dimensional vertical mixing model of the strait, and it predicts bloom timing pretty well for a simple model like that. So it's a vertical mixing model, and it performs really well for what it's intended to do. And it revealed some really interesting things, like how important wind is for bloom timing in the strait. So here's my model. I'm just kidding. This is metabolic pathways model of the human body. So this is not what we're setting out to do. I was thinking about this a bit yesterday, and it's like, do you have to look at a model like this to know it's a good idea to exercise or eat healthy? We can learn a lot about a complicated system with simpler models than something like this. So the processes we're interested in, I'm going to move through this. We're interested in processes that operate on decadal and the decadal temporal scale and kilometer spatial scale. And our research questions are related to the bottom-up hypotheses of the Salish Sea Marine Survival Project. So changes in circulation and water properties and how they may have altered the phytoplankton and zooplankton production and resulting in degraded food webs. Wind patterns may have changed, so that's really what we're focusing on, is how they may have influenced the food web and perhaps amplified up to juvenile salmon. In the beginning, we had Vili had a postdoc, Vijay, working on a pretty serious model based on URSIM. It was coupled to a hydrodynamic model called GETM. But that didn't quite work out, or we ran out of runway on that project. And subsequently, Carl and Joe and Vili have been helping to revive an old, simpler model called the Strait of Georgia, or what we're calling the Strait of Georgia Coastal Ecosystem Model. This is a simpler hydrodynamic model, and it includes it's a linearized two-layer barotropic model designed specifically to investigate how changes in wind, freshwater flux, and chemical properties promote primary productivity. The dynamics that are fast are modeled as time-varying equilibria and averages. Those that are slow are modeled as constants or forced parameters. The wind-driven horizontal currents and upwelling velocities develop after a few hours of steady winds. And so this SEM simulates nutrient concentration, turbidity, salinity, and advection fields, which are then used in Ecospace. Here is a simplified Ecospace ecosystem model. So emphasizing that we're really simplifying here, especially as this is sort of a prototype linking exercise between SEM and Ecospace. Ecospace is a spatially explicit ecosystem model that relies on the ecopath mass balance approach for most of its parameterization. And additional movement inputs are movement rates and estimates of importance of trophic interactions and habitat preferences and environmental responses for each of the functional groups. So this diagram shows the two zooplankton groups, predatory and herbivorous, and one phytoplankton group and one detritus cell. Juvenile and coho Chinook are shown here, but they've not been implemented yet. But they're the reason we're doing the modeling. So I wanted to show that we're using those groups, and we're modeling them as individual-based multi-stanza groups representing hatchery fish forced by external data on hatchery releases. I'm also interested in introducing young of year herring, but that's not been implemented. So these are linked via nutrient turbidity advection fields from SEM on the left. And those fields are combined with response functions. And then other data eventually, like light, that will drive primary productivity. And the primary production in Ecospace does draw down nutrients. So nutrients in the surface water should be depleted, and we should see bloom dynamics that are consistent with observations. Now, it's somewhat early in this initiative, and we are still working on things, but here is an image of some early findings. So there does appear to be different northern transport to a conversion zone, and that hypothesis does seem to be realistic given the advection patterns. So on the left, 1983, we see higher, red is higher, blue is lower, concentration of predatory zooplankton in the northern strait, as compared to June 2005, where the winds were were northwesterlies, transporting biomass to the south. We see a different level of concentration of zooplankton. So those salmon migrating north here in 2005 don't see necessarily as much food. Another finding here I thought I'd mention just interested me personally because it kind of tripped me up at first, and then I realized something's going on here that makes sense. So what I was finding is that just looking at the effect of turbidity on primary production seemed to have an effect on the primary production just outside the turbidity zone or the plume of the Fraser River. So on the image on the left, you can see a darker red color indicating higher biomass concentrations compared to in the turbid plume where primary production isn't really happening. But why is it so? Why is it higher than the northern strait? And when I turn off the turbidity, you see that when I turned off the grazing pressure, we see that effect isn't happening anymore. It's sort of simply more of a direct effect of the turbidity. So what's happening is actually, it does make sense. It's different rates of growth. You see, as the plume moves out of a certain area, the phytoplankton can respond quicker than the grazing grazers, the zooplankton grazers on those phytoplankton, and that leads to temporarily relatively high concentration right on the edge of the plume. So I thought that was pretty neat. But the status is that we're really still working on this model. We still have a long way to go. I've spent some time doing more of a rigorous evaluation framework than is necessarily commonly done on these models. But I thought it was important to know just how good the model is at predicting nutrients, especially in salinity in surface waters. So we're still working on that. Our next steps are to evaluate how the primary production patterns predicted by Ecospace compare to satellite chlorophyll data. And just wrapping up here, I'm running short on time. These are some of the tools that I've been using, and I really recommend looking at them. In particular, I really recommend using Jupyter notebooks, which help to kind of track in an interpretable way what you're doing with data, from the point it kind of enters onto your hard drive to goes into the model. I really recommend using GitHub Pages. It makes it very easy to launch little experiments in your browser using your browser as a GUI. And of course, you're probably quite familiar, most of you, with R and Tidyverse, but I find those to be very helpful. And one you might not know much about is, I don't find that people are aware of, is FME. That's a commercial software used a lot for data conversions, just to keep simple, especially geospatial data conversions, manageable. So I do recommend, if you're a student, you can get a student license for that, and we have, so that's been very helpful. Final thoughts, really simple. Simple's good, simplistic's bad, and it is a challenge to find the sweet spot. I had another demo I was going to show you, but I'll just keep moving. So just to rehash things, we have gone through several iterations of models: EcoPath, EcoSim, EcoSpace. I think that's just part of the process. We've prototyped an end-to-end model. We've prototyped an evaluation framework. We have a lot of data synthesis, and I do have a data management framework if anybody, other modelers, would like to discuss. So I think there's going to be more to come, though. So stay tuned, and yeah, thanks very much for your attention and attendance today. I much appreciate the opportunity.
Thanks very much, Greg, for your talk. It's a huge amount of information you've compiled and synthesized, and this is prepared for analysis. Yeah, so we have a couple of minutes for some questions. So I'd like to encourage, let's say, graduate students. We haven't heard much from graduate students in terms of questions. I'd really like to encourage you guys to speak up with your questions if you have some. So I'd like to, let's see if any graduate students or postdocs already have questions. I have questions of my own, but I'll leave those till later. So please speak up. Don't be afraid. You can also use the chat board if you'd like. If you'd like to put up a question there. Well, I'll start with one of my own. Greg, you were presenting information on the timing of various, let's say, events like herring spawning, and let's say variation in timing of like spring bloom, algal bloom, or zooplankton bloom, and so on. Is there much known about, let's say, the timing of migration of Chinook and coho into this Strait of Georgia? There must be, different stocks hit the seas at different time, Strait of Georgia at different time, and is there some information on that? And has that also been changing? We heard all about everything else in terms of how that is changing, but you didn't give us a picture on whether the Chinook and coho timing of migration into the sea has been changing.
Yeah, that's a good point. Murdoch. I didn't really get into that at all. There's definitely a whole suite of hypotheses and research done with respect to timing, and particularly looking at match and mismatch hypotheses. The hatchery, there's a bit of concern that the hatchery releases, the window of time in which the hatchery releases have been done has become more compressed over time. So releasing more fish over a shorter period of time in a bit of a pulse. And I don't have knowledge at my fingertips about specifically which hatcheries have changed their practices and which have not, or what the timing looks like specifically between coho and Chinook in different areas. But I know that there's a lot of research underway on that. And with respect to wild fish, I'm not sure a lot is known about that, but others on the call may know and could pipe up. But I think wild fish or wild juvenile coho and Chinook that are entering from streams and rivers are not well monitored compared to hatchery fish, although recent attention has been devoted and more research is being done on wild fish.
Yeah, it would seem that this mismatch sort of hypothesis in terms of say when the smolts enter the sea, let's say presumably the ideal thing would be that there would be a maximum of let's say abundance of good food types for them to forage on,
right,
when they're entering the sea. And like Carl said, that okay, there's this foraging arena hypothesis where if they have a lot of food there, then they can feed quickly, get what they need, and then work on avoiding predators. But if the food is sparse, then they're taking more risks and so on, a whole variety of other things. So that, like, have you attempted to sort of let's say test that sort of foraging arena type hypothesis in terms of there's an interaction of both predation risk and food availability sort of interacting?
Well, the foraging arena theory is really at the core of the modeling framework, and I didn't go into that, but I think I should mention it. That is an important piece of the puzzle for sure. It is of interest to test that. So since we have such detailed and long-term hatchery release data, we know when a huge quantity of fish are entering the strait, Chinook and coho, and their competitors are entering the strait. Carl has has calculated the number of wild fish that ought to be there based on survival trends and proportion of wild juveniles caught compared to hatchery juveniles caught. But the modeling setup is good. It's set up well to test these timing hypotheses once we have it tuned and up and running, and do intend to do that. So if we can get the bloom timing to be reasonable to the month, we can explore some of this match mismatch, those questions.
Carl.
Yeah, something that's been a puzzle about Chinook and coho is that they seem to have never eaten very much of the dominant zooplankton of the Georgia Strait, which are calanoid copepods. And the timing arguments, particularly about spring bloom timing, are primarily about mismatch arguments between callanus and a spring bloom and early callanus development stages. But it's not clear how that translates through to impacts later in the year when Chinook and coho enter the system a couple of months after that spring bloom usually. And they are feeding on weird things like one of their favorite prey outside of herring. Amphipods, black-colored pelagic amphipods and crab larvae and things like that. So that, and those things are basically, I don't know about crab larvae, but the most common thing, these amphipods are predatory. So we're looking at a shift up the food web as the season progresses to things that are feeding on the products of production from the copepods, and to expect some simple correlation between the bloom timing and so on and what ends up coming out in the trophic levels that these fish are feeding on 2 or 3 or 4 or 5 and 6 months later is a real stretch. And we don't see it in the clear correlations in the available zooplankton data.
Yeah, that's something I didn't cover as I intended to, is that specific, like explicitly what we're talking about being the postulated connection between the concentration of zooplankton biomass in the north and the juveniles. That's specifically the predatory zooplankton, the amphipods, that we're interested in modeling there. Which resist downwelling because they do vertical migration, active swimming in the water column, so they can avoid some of the downwelling that would occur in that kind of a hydrodynamic situation.
Even the algae avoid downwelling.
Greg, you'd mentioned earlier in your talk changes in wind patterns, and you'd indicated that, let's say, southeasterly winds would tend to make for higher densities of zooplankton in the northeastern part of the straits and vice versa. So did you actually test to see whether there was correlation between the spatial distribution of zooplankton and prevailing wind patterns within the Strait of Georgia, or is there no correlation?
We haven't tested the correlation using, say, sample data or the satellite imagery data that we have. But we do intend to look at how the model predictions fit what data there are. So no, we haven't looked at the correlations. One of the challenges with the evaluation is that, it's something that Jeroen Steenbeek is working on, so-called spatial temporal model fitting and evaluation. But we're going to try to use more typical statistics that, say, other modeling groups do use to compare with the chlorophyll A data we can get, the primary production data we can get. And there's good data for some of the later years from Monsieur Costa's lab at UVic.
Could you say something about, let's say, any relationship or interannual variation in terms of growth rates of Chinook and coho in the Strait of juvenile Chinook and coho in the Strait of Georgia, like growth rates and condition factor in let's say freshwater sockeye lakes? If let's say Lake Quesnel Lake, where there has been really poor quality food, the growth rates have been low and the condition factors have been low and the survival rates have been really low. So, are there any patterns there, like temporal patterns in condition factor and growth rates in juvenile Chinook and coho in the Strait of Georgia?
I know that there's been research. I know that Beamish and others have been looking at growth rates. I know there's some recent research being published on correlates of the growth rates, or the condition factor and the growth hormone or growth factor, and the abundance of zooplankton. So there's, I think, been, yeah, some relationships found. Yeah, definitely can kind of detect what's been a good year and a bad year. Yeah, but I don't, again, I can't speak to specifics.
We have long-term data just was published recently on the mean sizes of Chinook and coho in the commercial catches in the Georgia Strait up till '97 when the commercial fisheries were shut down in the strait completely. And there's been a major decline in the adult body sizes of older Chinook and coho in the strait. That isn't. There are weaker declines outside the Georgia Strait in some areas in average size, but really dramatic declines over exactly the period when the survival rates went down. So you're looking at coho that are by the 1990s weighed less than half at their second ocean summer of what they'd weighed back in the mid 70s. So there's still something's going on there, but we've also hypothesized that that could be due to increasing predation risk, driving them to reduce their foraging activity. They've shifted deeper in the water column kind of progressively as predation risk has changed out there.
Hey, Greg. Thanks. Great talk. I really enjoyed it. I just have a question. Talked about forage fish a lot, like herring and ulican. And I was wondering just how often they're available in Strait of Georgia, or do they migrate out? Like, are they there throughout the entire year? Is that why they're included in a lot of these discussions, or?
No, I mean, well, okay, so it is interesting to think of, most of the herring do migrate out of the strait and come back to spawn. They hatch in the strait, they rear as young of year juveniles, and then they leave in before the first winter. So, but then, I mean, we could go into the debate about whether there are some populations that tend to stick around all year, how big, those populations are in terms of biomass, and how maybe the eastern coast populations that have been not doing well in the last several decades and are starting to do better maybe tend to stick around. But by and large, I think most of the biomass of herring does exit the strait and then return as adults to spawn. But what we're concerned with here are juvenile fish that enter the ocean and are looking for food as soon as they hit the ocean in the first spring, summer, and fall. And at that time, they're there at the same time as young of year herring and some of the adults as well. So there are herring that these juvenile coho and chinook do see, and they go after. Ulican, I really don't know much about. Ulican have been a topic of recent research interest, and they are something that others could speak to, I'm sure. I believe they have a 3-year cycle. I believe they do leave the strait as well. I don't think they hang around. But I'm not positive about what their migratory cycle looks like. There's other forage fish of interest certainly as well, like sand lance. Not a lot is known about the trends in sand lance, but they are a really important diet component, especially for those fish in the San Juan Islands and Southern Gulf Islands. area too. So there's ongoing research there to try to figure out what the biomass is, and maybe they could detect some trends or figure some things out.
Gotcha. Thanks.
Are there any other questions? I think that yeah, we've got a long time overboard here, so it's 10:42. So I think perhaps, unless there are any other questions, we'll wrap it up here. So thank you very much, Greig, for your detailed presentation and